Welcome to McMasterminds, the show where we uncover the stories behind groundbreaking work and the incredible people driving it forward. In today's episode, we're honored to sit down with a postdoctoral researcher making incredible strides in the fight against cancer. Their work focuses on the substantial cancer research, tackling some of the biggest questions in the field to improve lives and advance our understanding of this complex disease. We are your hosts, Manat.
And I'm Diego. This conversation is one you won't want to miss. Let's get started and meet our guest, Dr. Emanuel Muruaga. How are you doing today, Emanuel?
Hey, hi guys. I'm doing okay. How about you?
We're good. Thanks for asking.
I'm happy to be here.
We're happy to have you here. Thank you so much for being here. It means a lot to all of us. So I think we could start by just talking about a little bit about your background, what is a postdoctoral? How did you get into this field or into the position that you're in right now? Do you want to just talk a little bit about your history?
Yeah, sure. Well, I did my PhD. As I'm from Argentina, and there's the educational system is quite different. So my degree, undergrad degree, is like a master equivalent here.
Mm-hmm.
And this is that was biotechnology focus undergrad. Then I start my PhD, and I did also a PhD focus in biotechnology and molecular biology.
Mm-hmm.
And then I moved to Canada to do my postdoc in cell biology. So I'm at SickKids working with a bacteria that's called H. pylori, and I think that we will talk more about that during all this session.
Yeah, definitely. So before we kind of get started, could you tell us a little bit about your transition? So you said you're from Argentina and you did a lot of your education there. What was it like getting your PhD and everything in Argentina and coming over to work in Canada?
Well, I think that the PhD life is more or less the same there and here, I feel.
Mm-hmm.
I mean, I didn't do a PhD here, but I see my colleagues that they are all doing PhD more. And I think that the life is more or less the same. Like you are very into research, for 5-6 years. In my case, it was 5 years
Mm-hmm.
and a month. And I defended my PhD and I moved to Canada 2 weeks after that to start my position here.
Mm-hmm.
Yeah, I was in a rush. Yeah.
Did you find it difficult to transition back to from Argentina to here? Was that a difficult move or?
Well, I think that there are different factors there because once you have your PhD, finding a position as a postdoc abroad,
Mm-hmm.
I thought it was going to be difficult. And if I see what happens with other life stories, it was difficult for other people. But in my case, I was blessed, I think. And I didn't have much difficulty to find this job.
Mm-hmm.
But I think that the point is connections.
Yes.
As in other sides of the life, if you have good connection, it will helps you a lot.
Mm-hmm.
And in science, it's the same. My PI back in Argentina had some friends here in Canada that were looking for postdocs, so they have money to pay a salary for a postdoc, and they were interested in someone.
Mm-hmm.
And I applied, so they interviewed me, and we had a match. So they liked me, they liked what I knew, what I was trying to do here.
Mm-hmm.
And actually, I did like what they were doing, because they were looking for someone to learn bioinformatics, because the lab is mostly wet lab based.
Mm-hmm.
So they don't have people running R or analyzing omics
Mm-hmm.
data.
Yeah.
So they wanted someone to learn that and apply it to their projects.
Mm-hmm.
So it was a very good fit for me because my background, as I said, it was more biotechnological and molecular based. So during my PhD, I developed a platform to purify proteins. You know that there are some already in the industry, but we were developing one that was cheaper. Well, so I didn't know anything about bioinformatics. I did art course during my PhD because I needed to do some to take some courses.
Right.
So it was interesting. It was the pandemic. I was at home, so I had the time to do like a long art introduction to art course. But I never used it like for my PhD actually. So I had that background that I never utilized, and then these guys were looking for someone with no background in bioinformatics to learn all the omics, how to analyze omics data. So it was a very good opportunity, I think, for me. So that's why I applied to this particular lab. So for me, like the transition was quite difficult and challenging. I mean, I was very enthusiastic because it was a really nice opportunity for me. But at the same time, it was a huge challenge because I know how to turn on and off my computer, but I wasn't very into coding and how to run all these omics data programs. So, and I didn't have any background in cell biology too, to know what's happening inside the cells. I was primarily like working with proteins, expressing proteins in bacteria or in some human cell lines. But I really never looked into what was happening inside the cell or the pathway, what's happening inside the mitochondria, what's happening with the peroxisome. So I didn't have any background in that. So when I started my position here, I was very like overwhelmed with all the information that I didn't know. And well, my project was all about that. So it was like a very huge learning curve at the beginning. I think that I'm still in learning a lot, but at least now I feel more comfortable with all the some cell biology. I'm not an expert, but I think that that's something that it's very nice about doing a PhD and a postdoc, because the main thing is that you learn how to be how to analyze data and how to be very critic to the science. So then you can learn everything else you can learn, but develop those skills is something you get during your PhD or postdoc. Or
that's actually very interesting because I think a lot of us, especially here, we come into our undergrad, with like, oh, we want to do pre-med or we want to do research and we want to do like, a very wet lab centered, we want to pursue that type of career. And within that, we always forget about the bioinformatics, biotechnology aspect of it. And that becomes really daunting. Like to this day, if I go through someone's research and they mention use of like Python or R, like you said, immediately I back off and I feel really daunted by that, or like it's very scary to me. But the fact that you like had very little experience with that and then learned from scratch by yourself in this position that you're in is so remarkable to me. Completely crazy. But you talked a little bit about like how your content from what you learned, like the science content that you learned in Argentina kind of translated over to Canada. But what about the culture? Did you think like the science community works and operates differently in Argentina versus here? And like, just the communication part of it, how's like the dynamic in a lab different, or is it different at all?
I think that, for me, it was also very challenging the language because I was used to speaking Spanish all day. And of course that I took some classes for English, but here it was all English.
Right.
So that was very hard. I think that I'm still struggling with that, but I'm getting better.
Right.
And regarding the lab culture, I think that I feel that here, as we are exposed to like very high quality science. So I feel that the pressure and the standards, at least for me, are higher. And that's something that I feel it's very cool because I want to be at that level, and I feel that I'm pushed to have that level. Back in Argentina, at least in my lab, the science is not very basic science. mostly applied to biotech companies.
Mm-hmm.
So it's more that I also like that it's more about developing techniques and not basic science. So you take everything that is already known about a particular disease or whatever,
Right.
and you develop a low flow device to detect that pathogen or that bacteria, or in my case, to develop a new purification system for proteins that is cheap. I feel that, of course, the culture also changed a lot. So of course, that I felt more comfortable back home because it's my own culture. Like,
Right.
I understand everything, the small. Yeah, like here I'm still a stranger, still a stranger. So, but I think that that's also an opportunity to grow because you are exposed to different cultures, different food. Like Argentina is a very homogeneous country, so we don't have a lot of different religions, color, races, or
Right.
whatever,
Right. Yeah.
even food. So I think that's something that is nice about Canada, that you are exposed to very diverse environments. Yeah.
Nice. So, you mentioned that you do research regarding H. pylori. Would you
Yes,
mind
yeah.
giving a little bit of a breakdown? We'll talk about your specific project in a minute, but kind of for the people who may not know what H. pylori is, could you give kind of like a breakdown, what it is, where can we find it, kind of the whole H. pylori breakdown?
Yes.
Where can we
Well,
find it?
in your stomach. I hope not, but yeah. So H. pylori is a bacteria that it's inside us in our stomach. It's a pathogen, so half of the population is infected with this bacteria. Not all of us have the bacteria, but it's a very common bacteria to find in our stomach. There are different types of H. pylori. They are like relatives, and some of them are very bad for our stomach. So if you have H. pylori, some strains of H. pylori, you will develop gastritis maybe, or ulcers in your stomach, and that can also lead to gastric cancer. So 90% of gastric cancer, how do you say that? Like the people that has H. pylori, sorry, 90% of the people that has gastric cancer are related to have been infected with H. pylori.
Wow.
So it's a very high risk to develop gastric cancer. That's why most people in the gastric environment are doing research in H. pylori now, because the risk to develop gastric cancer is very high. So we are not a lab that do that research cancer, but we are trying to link like H. pylori infection with the development of gastric cancer for sure.
Yeah, so the bridge between the two is what your lab focuses on.
Yeah, exactly.
So like when you say that H. pylori is in the stomach, right? We know that the stomach is like a very harsh environment. It's not a fun place to be in. So what makes H. pylori kind of different compared to other bacteria? Like how does it stay in the stomach and colonize the stomach?
Well, you know that inside us we have a lot of bacteria that live with us. It's our microbiome, and it's getting more like people is getting more interested in knowing how that affects us, because it's almost like it's like part of us. It's an extra organ. But as you mentioned, Diego, like the stomach is a very specific and difficult environment to live, because it's like a pool of acid. So for the bacteria, it's very hard to live there. So bacteria mainly lives in the intestine, but these bacteria developed some tricks to live inside the stomach, this very harsh environment. So basically, it has a molecule, is a toxin that transforms the urea in ammonia, and that's like makes a shield around the bacteria. And then you know that in the lining of the stomach, we have a thick mucus layer that is protecting our cells from that acidic environment. So the bacteria goes into the mucus and can live there. But you also know that maybe that the mucus is constantly turning over. So the bacteria also has some molecules in the surface that allows it to attach to the cells in the epithelium. So basically, I think that that's how the bacteria manage to survive inside us. And something that is very interesting from my lab is that a few years ago, they discovered that actually the bacteria can go inside some cells that are called parietal cells. So it's a cell type inside the stomach. So another way that how the bacteria is surviving this environment, and also the antibiotic treatment is living inside our cells.
Yeah. So then, do you think that has something to do kind of with how H. pylori evolved to stay in the stomach?
Well, I think so. I think that there are no much risk. I mean, we cannot fix reason to evolution because it just happens. I think that for the bacteria, it was an environment where the bacteria didn't have many competitors, maybe. And there are a lot of good resources because you have all your, well, the mucus is a very rich environment for the rich amino like food for the bacteria, and also with all the food that we are incorporating when we eat, it's also giving the bacteria material to live. So yeah, I think that that's how maybe the bacteria evolved to stay in the stomach.
Right. Okay. So I think for our listeners, they would probably have a good understanding of what H. pylori is at this point. So I think now might be a good time for us to talk about your specific lab. And so do you want to preface what your lab is first before we dive into some of the deeper questions? Very specifically about your lab.
My lab. Well, my lab is a very small lab actually now. So my PI is a gastroenterologist, so that's why she's very interested. It's called Nicola Jones. So she's very interested in H. pylori. I think it's one of the scientists more relevant in the H. pylori field, internationally meaning. And there is also another research associate and then me. And we are all researching different aspects of a toxin in particular, because H. pylori has been known because of one toxin that is called Kage, that is the one more related to cancer. But it also has another toxin called Bacche, that is the one that allows the bacteria to penetrate the cells that I told you recently. So all the work in the lab is about this toxin, mainly Bacche, and how this toxin is transforming the epithelium to develop different diseases.
First off, are the toxins different strains or different, like, what are the toxins when you talk about H. pylori? Like, what is it, just something that's inside of them? Just for clarity for myself.
Yeah, no, the toxins are something that is secreted by
H. pylori.
So H. pylori, yes, H. pylori generates inside the bacteria, because you know that the bacteria inside is like a fabric, so it generates the toxin inside, but it secretes the toxins, Kage, Bacche, or there are many others, to produce a toxic effect in this case in the epithelium of the stomach.
Oh, okay. That makes a lot more sense. And so this is not something that can be treated with antibiotics, and this is not something that we can kind of deal with in different ways.
Well, yes and no, because, I mean, if you want to kill the bacteria, you should need antibiotics. But now because of the research in my lab, it's known that if you treat with the bacteria, you will kill everything, but the bacteria that are inside the cells are not being killed. So the treatment actually is a cocktail of some antibiotic, because also the bacteria, you know, that a worldwide problem is the resistance to antibiotics. So the bacteria are mutating very fast, and as we are treating with antibiotic, they develop resistance to the antibiotics. And H. pylori is the same case. So H. pylori develops resistant to some antibiotics. So now you have to take a cocktail of antibiotic for like 20 or 30 days,
right,
to get rid of the bacteria. But the toxin is another case. I think that there are no, at least my knowledge is that there are not treatment focused on the toxin.
Okay.
But toxin over time is being degraded, so it will have like a very harmful effect in your stomach, but then it will be clear.
Yeah.
Got it. Okay.
So then, in that case, what else do we really need to know to kind of further, like you said, we have this kind of problem where the bacteria that's in the cells are not getting hit, and we kind of are trying to find, I guess, a better way of treating this. What else do we need to know to kind of advance the treatment?
Well, I think that there is too much information that we don't know about the bacteria.
Yeah.
Because we treat bacteria as they were all the same. So we treat with, I don't know, metronidazole, like it's a very common antibiotic.
Right.
So it will kill a wide range of bacteria. But if we want to target one specific bacteria, we should need more research about the specifics of that bacteria. So I think that for treatments, we need more research about knowing all the how the bacteria manage to live there. We know something, but we don't know everything about the bacteria. So focusing more on that for sure will give us more clues on how to get rid of the bacteria. For example, my lab, once they discovered that they have this intracellular bacteria, they start developing new drugs that they are not antibiotics to target those bacteria that are inside. Because the problem is that if you kill all the bacteria that are outside the cells, once you discontinue the treatments, the bacteria that are inside go out and recolonize the stomach. So that's the problem. Like you have like a how do you say that? There is a specific word for that. That now I don't remember, but you are being recolonized.
Right. And so you mentioned, okay, so for a lot of us that are listening today, they're little scientists, so we kind of know how antibiotics work. You know, a lot of our course content that we, teach in university and learn in university talks about antibiotics. But you mentioned some of the other drugs that are not antibiotics that are kind of focusing on, getting rid of this toxin or rid of these bacteria. Can you maybe talk a little bit about how they do that? Like the scientific, biological way they function when they go inside?
That's a very good question, but because I really don't know the answer.
I got you stumped.
Yeah, I never work with. There are some. I know that there are some antimicrobial peptides, but I don't really know how they work. And for example, we are. My lab collaborates with a biotech company in Boston, so the biotechs know some compounds that are used for this treatment. So they don't disclose which molecule is. So you really don't know what they are. For sure, there are many that we know what they do, but in this case, we don't know even what's the chemistry of the molecule. We just have like a very wide name, and that's all. But well, this is a very specific case because it's a compound that is being patented, so that's why the company doesn't release like all the information about it.
Right. Honestly, this just goes to show how, like, how nuanced the science world is. Like, a lot of us, we just see, oh, a new drug came out or there's a new therapy style. But then we don't realize how many different labs from how many places in the world all had to work together to, like, produce this one singular thing that we see. So, honestly, again, very
Yes,
remarkable
that's
to
true.
see that. And it must feel so good to be part of something like that, no matter how big or small the part is. You know what I mean?
Speaking of
Yes,
that,
it is. Mhm.
you mentioned, so you've been kind of sharing with us what your whole lab collectively you guys focus on, but could you tell us a little more about what your specific focus is and what your specific role is in all of this?
Right.
Yes. So I mentioned that there is this very particular cell type in the stomach that are called parietal cells. So the bacteria we know that enters the parietal cells, and with time, the parietal cells, if you have a very chronic infection, they will die in your stomach. And these cells are the ones that produce the acid environment. So it has some proton pumps, so they release these hydrogen molecules to the stomach, and that's why we have an acidic environment there. So the stomach, the bacteria, we know that lives inside these parietal cells, like transform somehow all the pathways inside the parietal cells to get rid. In one hand, they get rid of the parietal cells because in the long term they kill all the parietal cells, but at the same time the bacteria transform all the epithelium because it's like a chronic injury. So there are many other cell types that are interacting there and are trying to repair your system, and that we don't know how will promote cancer. So that's what my research is focusing now. I'm working with omics data, so it's a very huge data amount of data because I have I don't know if you know single cell RNA sequencing, but basically is that you sequence everything inside all the transcript, the mRNA transcript. So you know what is happening inside each cell inside the stomach. So then you have a very huge amount of data that now I'm trying to analyze to see. So I infected the mice with the toxin and with the bacteria with the toxin and without the toxin. So I'm trying to compare the two conditions to see what is the toxin particularly doing to the parietal cells that leads, and what's happening inside the parietal cells in particular, because with this technique, you can focus in different cell types. I mean, my research will, in the close future, move to the immune cell type, because we know I mentioned that the bacteria cause ulcers and you have gastritis, so you have a lot of inflammation in your stomach when you have the bacteria, and that means that there are a lot of immune cells involved. So we would like to focus on that, but for now I'm focusing only in the parietal cells to trying to see what the toxin is doing inside these cells and how these cells are trying to survive or to reprogram, and that's basically what I'm doing. Yeah.
Okay, so again, this is all types of like the little words you say here and there. They get me so excited because I've been, learning about this. But that's all just, textbook knowledge. But to hear that like this is real stuff that's happening and not just like theoretical stuff that I have to read once and forget about it, is really interesting.
And that wraps up the first half of our conversation. So far, we've explored the fascinating science behind our guest work with H. pylori, but next episode, we are shifting gears. We'll dive into their personal journey, the challenges, the triumphs, and what it really took to go from a high schooler from Argentina with big dreams to a postdoc making a massive impact. Stay tuned, you won't want to miss it.